Good morning, everyone, and welcome to today's MPG Primer. We are very excited to have Ben Strober with us today. He's a postdoc in Alka's Price's group at the Harvard T.H. Chan School of Public Health. He works in statistical genetics and particularly on the integration of GWAS and functional data to better understand the molecular underpinnings of disease. Prior to his postdoc, he got his PhD with Alexis Battle at Johns Hopkins, where he focused on the context-specific regulation of gene expression. And so today he's going to tell us about his work advancing our ability to interpret GWAS loci using molecular QTL data. He's glad to take your questions during his talk, so please feel free to walk up to a microphone, raise your hand, and he'll answer your questions as we go. Thanks so much.
Thank you for being all here so early. Today I'm going to give kind of a broad overview of this space of using molecular QTL data to try to interpret the biology underpinning GWAS loci. So to start, I'm going to start with some motivation. Why do we want to do this? And then introduce quantitative trait loci in general. What are they and how do we discover them? And then kind of get into the meat of the talk, which is going to be on kind of integrating these two first sources of data and talk about several approaches for integrating this GWAS and QTL data and what we can do with it. And then lastly, talk about limitations of such approaches of using QTL data for interpreting GWAS loci. But yeah, I mean, this is meant to be a general overview. So if people have questions, definitely feel free to interrupt me and let me know. But to start, what is the motivation here? We want to interpret GWAS loci, and you know, and I hope, I imagine this is background for a lot of people, but GWAS over the recent years has been shown to be able to identify hundreds to thousands of loci, independent or nearly independent loci associated with a disease or trait. And here we just show the number of loci identified for 3 representative traits: BMI, height, and waist-hip ratio adjusted for BMI. As we vary the sample size, and we get kind of increasing number of hits as we get at larger sample size, and one can imagine you end up getting to these truly very polygenic traits where you're having hundreds to thousands of loci associated with disease, and you can imagine getting Manhattan plots that look something wild like this, where you're getting tons and tons of hits, and the question is, what do you do with them, and how do you understand them? And I think largely this is largely the biological mechanism underlying a substantial fraction of the disease loci remains unresolved. And, in this talk, we want to ask the question, take this locus, and perhaps this is a bad example because perhaps maybe someone's figured out what's going on here, but I'm going to imagine we haven't. And we want to know, what is the mechanism underlying this locus? And, biology is complicated, and, we want to try to boil it down to simple things, but you can imagine thinking your trait variants, and from your GWAS with your very large sample size GWAS, that this trait variant is associated with the trait, and you want to figure out how it happens. And people have kind of looked into this, and kind of a couple of the main ways in which a trait variant can go ahead at the immediate molecular level to have an influence on trait. One thought would be it goes through regulation of gene expression levels, so total mRNA levels. The variant causes the mRNA to go up or down. And alternatively, we have this row on the bottom where instead of the variant causing the gene expression to go up or down, perhaps it causes a splicing change, and you get a different isoform existing for the gene, and therefore that has a downstream impact on the complex trait. But ultimately, as the very large overview of this talk, what GWAS is going to do, it's going to identify these SNP disease links, which we have here. But we have this question is what is the biological mechanism? And that's kind of a vague statement, but for here, or for the purpose of this talk, I'm going to view kind of the biological mechanism as, what is the gene or potentially plural genes of action? Is the variant acting through splicing or modulation of total expression levels? Is there a tissue or context that gene is acting in? And basically, to answer this question, we're going to need some additional source of data beyond the GWAS data, and hence queuing in QTL or quantitative trait loci. And a QTL, I don't know if there's much need for a distinction between a QTL and GWAS, but a QTL is just an association across individuals between a genotype of a particular variant and the expression level of a particular gene. Well, I meant this to be general. So a QTL can be any molecular phenotype, not necessarily expression level of a particular gene. A specific type of QTL is an eQTL, which is where the QTL affects the expression level of a gene. But in general, a QTL is just like a GWAS, but for a molecular phenotype. And here we have individuals stratified by genotype of a particular SNP on the x-axis, and we're showing their value for the molecular phenotype on the y-axis. And this is clearly a QTL, meaning the variant we can believe has some regulatory effect on the expression level, or this is expression of the gene. And there's a whole bunch of different types of molecular QTL, and I think there could be a whole bunch more. If people decide to generate these molecular phenotypes, and also in population scale level data, and also get genotype data for those individuals as well. But I think in my eyes, the most widely popular ones are eQTLs or expression QTLs, where you have variant affecting expression or total expression levels of genes. Splicing QTLs, where you have a variant affecting the splicing patterns or the isoforms that exist. Then you can also have pQTLs, which is variant directly affecting protein levels as opposed to mRNA expression levels. And then, the list kind of goes on and on: chromatin accessibility QTLs, DNA methylation QTLs, and so on. And so, you can imagine kind of this wide source of data where you're able to get these variant to molecular phenotype mappings in addition to these variant to trait mappings. And one additional distinction that I'm going to talk about is this idea of cis versus trans eQTL. Cis, it's just kind of a subtype of considering which variants to test for association with a given molecular trait. And a cis eQTL is going to limit to variants within a certain small window around the given molecular element. So if you're thinking about an eQTL or an expression QTL, for a cis eQTL, perhaps you're going to limit to variants within a 1 megabase region around the transcription start site of the gene, whereas a trans eQTL, if you're on the same chromosome, you're going to limit to variants beyond a 1 megabase region around the gene, and in addition, consider variants effect on that gene from other chromosomes. And I think this distinction of cis and trans is done in general because basically the genetic architecture of at least expression molecular traits is kind of very cis versus trans dependent, in that there are these kind of very large effect cis effects, and then there's kind of a more polygenic GWAS style trans effect. And from a consideration of statistical power, it's often useful to consider just cis eQTLs by themselves. And so for kind of the purpose of this talk, I'm gonna limit ourselves to the cis eQTL category. In addition to that, I'm gonna kind of mostly talk about expression quantitative trait loci, as that's kind of, I think, where the most data is at the moment. But in general, everything on the methods I'll be talking about are going to be applicable to kind of this broad range of molecular phenotypes. And here we're just showing an example of QTLs. And in general, the point I want to make is that people have generated eQTLs at this point in a kind of a vast array of tissues. And even nowadays, with the advent of single cell data and using it to generate single cell eQTLs, basically eQTLs in specific fine-grained cell types or context. And here we're just showing plots from the GTEx consortium, where they identified eQTLs in around 50 different human tissues, and basically just showing that we get thousands of genes with significant eQTLs in basically all these tissues, and presumably with more samples you're going to get even more, as we kind of have this linear dependence of eQTL detection here with sample size, and so these genes in general are these very heritable molecular phenotypes that we're able to measure, and they have similarities across tissues, but we get kind of these independent or somewhat independent sources of information across tissues as well that we can ultimately use to integrate with GWAS data. And here's kind of just my slide motivating the use of QTLs for use in interpreting GWAS loci. I mean, I think we have reason to think that it makes sense that if a variant affects a gene and the variant affects a trait, perhaps that the variant might affect the trait through some regulatory mechanism related to the gene. And here we're just showing two example analysis, one from Yang Li et al. Science 2016, and one from Alkas's group in 2018, just showing that QTLs, in this case, on the right side, it's just limited to eQTLs, but on the left side, they're looking at a whole bunch of different classes of eQTLs, SQTLs, eQTLs, and so on, and basically showing just that there is considerable overlap in the loci that are identified as eQTLs and the loci that are identified as GWAS hits. But this analysis shown here is kind of from a global genome-wide perspective. Can we see enrichment or kind of broad overlap of QTLs within GWAS loci? And what I'm going to talk about today is on a more locus level kind of picture. So zooming in and seeing how can we integrate eQTL and GWAS data to uncover the biological mechanisms at a disease loci. And so in general, these eQTLs are going to generate these SNP gene associations. We'll have these list of SNP gene associations. And these GWASs are going to identify these SNP disease or trait associations. And ultimately, we want to kind of link these two together, and hopefully you can get at these SNP gene to disease trait kind of pathways or potential biological mechanisms. So that's kind of the hope for here. And so now I'm going to kind of shift gears a bit and talk a bit more methodsy and talk about potential approaches that people have taken over the past, I guess, decade at this point in terms of integrating QTLs and GWAS data, and what are their limitations, and how have we kind of advanced since then, and ultimately I'll talk at the end of where we're at now and what are the current limitations of state of the art. But first, I'm going to kind of give this broad overview of approaches for integrating GWAS and QTL data. And I'm going to kind of lump these into two categories. And first are kind of these initial, but very widely used, and I'm going to call them quote unquote single gene approaches for integrating GWAS and QTL data. And namely, these are quite popular, COLOQ and TWAS, or transcriptome-wide association. And what I mean by single gene is basically these approaches consider each gene's effect on the trait in isolation and don't consider the fact that there are potentially genes nearby each other that are related, and one gene may potentially tag another, leading to false positives. But I'll get more into that later, and I'll talk about these kind of approaches people have taken to mitigate these problems, which I'm going to call multi-gene approaches. But to start, I'm going to introduce these kind of initial approaches for integrating GWAS and QTL data, and the first is this really nice and clean approach called COLOQ, or colocalization, which basically just calculates the probability that a variant is causal in two association studies. And I think it's widely used for where one of those association studies is a GWAS and the other association is an eQTL study. And here we just have a really nice example where we have a given region of the genome, and there's kind of a clear peak in the GWAS Manhattan plot, and there's also kind of an equivalently identical peak in the eQTL study. And what COLOQ is going to try to do is calculate some posterior probability on whether or not the variant that is causal in the GWAS study is identical to the variant that is causal in the eQTL study. And so that's really all it's going to do, and it's going to kind of additionally make this assumption that there's one causal variant in the region. And that's going to be a limiting assumption, but in practice, if it's not a super polygenic trait, maybe it's perhaps kind of reasonable, but there's going to be approaches we're going to talk about later where people extend this and talk about more complex approaches. But I want to introduce this because I think it's kind of a fundamental method, and I think it's nice and elegant and easy to understand. But what COLOQ does, kind of under the hood, is it calculates the probability of 5 different, what I'm going to call causal variant hypotheses in the genomic region, and it's going to assume a single causal variant in each study, or it's going to assume at most a single causal variant in each study. And basically there are these 5 hypotheses, and in their space, the sum of the probability of each of these 5 hypotheses has to sum to 1, meaning basically these hypotheses capture all the possible events that can occur in terms of the causal variant. And the first is, well, there's no variant associated with either trait. That's hypothesis 0. The second is hypothesis 1 is the variant is associated with trait 1, but not trait 2. And the third, I guess, my index is weird, but the index 2 hypothesis is the variant is associated with trait 2, but not trait 1. H3 is the variant is associated with both trait 1 and trait 2, but you have a different causal variant between the two. And the fourth, which is, I think ultimately the one they're interested in here, is you have the same variant associated with both trait 1 and trait 2. And they're going to basically call this fourth hypothesis colocalization. And ultimately, coloc is going to calculate a posterior probability on H4 being true. And how does, okay, so I guess a bit more in details. Kind of visualizing these four hypotheses and how can it occur. And so this graphic here is going to assume there are n variants in the region. And these n variants are these little circles here. And so for hypothesis 0, there is basically no causal variant in either study. And this is basically one variant configuration. It can only happen one way. There's no causal variance. And the second one is there's a causal variant in the GWAS study 1, which is depicted by these red circles. And in this case, the causal variant could be the first SNP, the second SNP, the third SNP, or so on up to the n SNP. So there's n possible ways for this to occur. And then equivalently for hypothesis 2, in which you have a causal variant in the second study, but not the first. In this case, there are, again, n possible ways for this to occur because that single causal variant only in the second study can be at one of the n possible variant positions. And then there's H3, which is kind of getting more interesting, where you have a causal variant in both the red and the blue study. And so in this case, but you have a causal variant in both the red and the blue study. But in this case, specifically, the causal variant can never be the same. And so in this case, there are n times n minus 1 possibilities. And those additional n possibilities here occur from the causal variant being the same. So these are kind of all the outcomes, and coloc is ultimately going to kind of calculate a probability for each one of these kind of 1 plus n times n minus 1 plus n different events occurring, and then kind of tally that up and get an ultimate posterior probability. And in more detail, they're going to call this probability of interest PPH4, or basically the probability of hypothesis 4 given the data. And you can just rearrange this by dividing by the 1, basically, where P of H0 plus H1 plus H2 plus H3 plus H4 equals 1, because as we know, all the hypothesis basically encapsulate all the possible outcomes, and therefore it's probability 1. So dividing by 1 is no big deal and changes nothing. And then furthermore, they divide both numerator and denominator by H0, probability of H0. And so at this point, we've done nothing other than kind of rearrange some terms. What it allows for them to do is have these terms now. Can people see my cursor? Yeah. Basically have these terms, basically the probability of H4 given the data over the probability of H0 given the data. And what this ends up working out to is it's basically this posterior on a likelihood ratio. And it works out really nicely in that for a given P of HK given the data, normalized by the null, which is P of H0 given the data, all you have to do to calculate that is sum over all the possible configurations in state K. So say state K is hypothesis 4, basically. One configuration is variant 1 is causal in both. The second configuration is variant 2 is causal in both. So in this case, sum over n possible combinations. And all you have to do is calculate these couple things. And this right side is the prior, and that's user specified. So they don't really have to worry about that. And this left side is just a likelihood ratio, and it ends up being just this relative support for SNP K being causal in trait 1 and SNP J being causal in trait 2. And what this works out to is just a product of Bayes factors. And for those familiar with fine mapping, Bayes factors are kind of these statistics that quantify the relative support for a SNP being included in the model. And what's really nice about Bayes factors is you can calculate them entirely from GWAS or EQTL summary statistics. Meaning here, they're able to encapsulate, all these posterior probabilities just using summary statistics and marginal summary statistics and marginal EQTL summary statistics. But so this is kind of my broad overview of coloc. And in general, if you have a single causal variant in a region and you want to see whether or not that single causal variant is shared between two studies, I think this is a great approach. But the problem is, as we know, it makes a strong assumption that there is a single causal variant. And so now we're going to move on to a different approach, which has a lot of parallels to colloq, but does it a bit differently. And it's called a TWAS, or a transcriptome-wide association study. And all a TWAS does is it calculates the marginal association between the predicted genetic gene expression and disease. And so what do I mean by predicted genetic gene expression? Well, it's a two-step approach. And in the first step, you take your EQTL data, where you have, kind of hundreds or so of individuals of people where you have paired variant genotype and expression data, and you build some type of predictive model predicting the gene expression from the variants. And so in this case, basically, the predictive model is saying that there's a certain amount of weight for variant 1, and there's a certain amount of weight for variant 5. And so you build this predictive model from your EQTL data. Then in your GWAS individuals, you predict what their expression is. You get predicted expression in your GWAS individuals. Then with that predicted expression, you associate whether or not that predicted expression is significantly associated with the disease or trait. And from this, you kind of get a marker of whether or not the expression level of the gene is associated with the trait, with kind of the caveat that we're not really talking about expression, but we're talking about genetic gene expression. So it's kind of the subset of gene expression. And even more so, it's not even genetic gene expression, it's predicted genetic gene expression, which is going to be limited by finite EQTL sample size. But so this is kind of the general approach. And the early work did some really, in my eyes, smart things to allow that you don't actually need the individual level GWAS data to perform this analysis. As I described it, you actually, you get your predicted gene model and then predict in the individuals in the GWAS, which is, what the model intends to do. But they've done some kind of clever statistical tricks to allow this same association to be approximated using just GWAS summary statistics and a reference panel of LT. And so you're able to approximate this kind of true trait association of predicted expression with the disease or trait, just given your predicted gene model for the EQTLs and given GWAS summary statistics and some reference LD panel. Okay, so I've introduced these two methods, TWAS and colloq, and how are they similar and how are they different? Well, TWAS, what it basically does is estimates the genetic correlation between genetic gene expression and the trait, or correlation or association. It's kind of this, it's a frequentist approach where it gives you a p-value at the end of the genetic gene being associated with the trait. In addition, it requires no assumption on the causal variance in the model. It assumes, whatever type of predictive model you use to fit the gene, that's what assumption you're making on your data. That's what variant assumption you're making. So if you use, say, a lasso model to predict your gene expression in the EQTL data, you're making this assumption that the variant effects on the trait and ultimately the disease are sparse. But that's it. You can have multiple causal variants and so on. And basically, depending on what type of predictive model you do, it's going to allow for really rich predictive models. Colloq, on the other hand, is estimate the probability of a single shared causal variant between your expression and the trait. It's kind of this Bayesian approach, and it's going to give you basically a posterior probability ranging between 0 and 1. It has a strict assumption on the number of causal variants, which is 1. I mean, there's been some work that extends it to slightly higher, but in general, people use it as 1. And it basically just uses marginal summary statistics from both the GWAS and the QTL study. Okay, so now I'm going to shift gears a bit, and I've introduced what I'm going to call these single gene approaches, and I want to talk about their limitations and kind of move us into more modern work on integrating GWAS and QTL data. And what do I mean by this single gene assumption? Both TWAS and colloq basically consider a gene trait pair in isolation, so it considers a single gene at a time. And analogous to variant linkage disequilibrium obscuring tagging variants from causal variants using marginal GWAS associations, A significant GWAS association for a given gene can result from one of two possible things. Well, the first is that the gene actually has a causal effect on disease. That's one outcome. Another is that the gene does not have a causal effect on disease, but is simply correlated or genetically correlated with another gene that does have a causal effect on disease. And here we just kind of exemplify that point in a schematic from Mancuso et al., Nature Genetics 2019, where we basically show that nearby genes can exhibit high levels of what I'm going to call genetic co-regulation, but that's a fancy word for just correlation amongst the gene-gene pairs. And we first motivate this, or they first motivate this, from a GWAS style plot, where we basically showed the Manhattan plot of the negative log 10 p-values for variants associated with a trait. And basically, because of linkage disequilibrium, which we're showing by this LD matrix on the bottom, you're going to get basically a whole region of SNPs that look like they're associated to the trait, just because perhaps there's just one causal variant that has an effect on the trait, and the other ones are correlated with that causal variant and look significant. Well, what they're saying here is the same thing can happen for GWAS associations. If you have a given gene that is causal for the trait, and that gene is correlated with other genes, all those genes in that correlation block are going to look associated for the trait. And this can lead to a number of false positives.
Then there was one question from online, which was, sorry for the last slide more so, but do you find that GWAS and colloq results generally overlap, or do they overlap in the interesting regions, or, kind of in your experience?
I don't know if I have great experience to answer that. I do know that Sasha Gusev's 2016 paper, which is cited, Gusev et al., Nature Genetics 2016, has a pretty explicit comparison, I think, in simulations of colloq and GWAS. And then I think there's one other from there's a alternative to GWAS called PredictScan, and I think they also have a pretty extensive comparison. In general, I think, they're, particularly if you have a single causal variant, they're going to lead to highly concordant results. Once you break the single causal variant assumption, it ends up getting different. I don't have a great answer in terms of like FDR and power if you have like a single causal variant, but my recommendation would be to look at this 2016 paper.
Thank you.
Okay, so genes in isolation or not, and this paper from Nick Mancuso in 2019, I think really highlighted the importance of this, and that really similar to LD, I think kind of initially plaguing GWAS studies, correlation between genes, is really going to plague GWAS studies, or any type of variant to gene to trait style association. And here, we're just showing an empirical gene-gene co-regulation or correlation matrix, a whole bunch of genes in a given region. And what you can see, is that, there's a whole bunch of these really high kind of co-regulated or high correlation blocks here. And what you can imagine, like, say, hypothetically, this gene here is causal for disease. All these guys here are going to look significant. These are going to look, well, maybe I got that wrong. Perhaps this will look significant. And so how does this occur? I mean, co-regulation between two genes can occur from basically, in my eyes, two phenomena, maybe more, but I think it's just two. First is that two genes have the same causal variant, which is completely reasonable, and I think there's been strong evidence showing that this happens sometimes. But I think the second more predominant thing is that two genes can have different causal variants, and those different causal variants are just in LD with one another, which will cause those genes to look like they are correlated with one another. And so how do people initially aim to kind of tackle this problem? Well, what I think people have been focusing on in the past years is taking a fine mapping style approach. And this is really analogous to the variant linkage, using fine mapping to disentangle causal from tagging variants in a genome-wide association study. You can use a fine mapping approach to disentangle causal from tagging genes in a GWAS study. And how do they do this? And so this is summarizing the method FOCUS, which was presented by Mancuso et al. In Nature Genetics 2019. And FOCUS takes a two-step approach similar to GWAS, in that in the first step, for each gene, you're going to estimate predicted genetic gene expression for each gene using EQTL data. And so for, you have this panel of genes and you get the gene model for each gene. And the second step, they basically find map genes or identify genes with non-zero effect on disease while accounting for other genes. And I won't go into the details of find mapping because I think it's kind of beyond the scope here, but basically, find mapping in a brief summary is this kind of a multivariate model that considers a whole bunch of predictors in combination. In this case, the predictors are all the genes and assumes basically specifics, very sparsity-inducing prior to identify a small subset of the predictors that are actually causal for the outcome. And so in this case, you're considering all these genes in competition and basically trying to disentangle the co-regulation that occurs between genes and try to, pinpoint what the causal gene is. And so in this case, hypothetically, gene 2 and gene 5 are find mapped as being causal for the disease. And lastly, I just want to highlight, there's been another recent work in this space, Zalidol Nature Genetics 2024, which just came out, in which they're calling causal GWAS, and it kind of just extends the focus work, this gene find mapping work, one step further by saying, well, look, like a gene, could be falsely associated with the trait because it's correlated with another gene, but in addition, that gene could be falsely associated with the trait because it's correlated with a genetic variant that is not in this assayed gene pool. And maybe that variant goes through some other regulatory mechanism that's not a gene. And so in this case, they basically jointly find map variants in genes and basically show that, if you have a simulation setting where you have basically some effects of variants that don't go through the assayed genes, it's really important to consider, this kind of joint model of these quote unquote, I'm calling them non-mediated genetic variants and genes in this joint find mapping model. And so this last one is called the causal GWAS. And in this causal GWAS paper, they show this really nice example, kind of exemplifying the benefit of not considering genes in isolation. And so here, what they're showing is basically a joint Manhattan plot of the GWAS associations and the TWAS associations. And the GWAS associations are these little circles, and the TWAS associations are the squares. And so what you can see is you get a whole bunch of kind of significant variants associated with the trait, or a whole bunch of marginal associations between variants in the trait, and a whole bunch of marginal TWAS associations between genes in the trait. So for example, you're seeing gene 1, gene 2, and gene 3, looking quite significant TWAS in terms of significant. And in addition to that, this gene 3 and gene 1 are both identified via co-localization with posterior probability 0.995 and 0.998. So, 3 of them looking significant by TWAS, 2 looking significant by colloq, and then you kind of apply this joint find mapping and it's able to tease apart, all these variants and all these genes and say, look, this guy here, this gene is your causal one, and the other ones are just along for the ride and tagging the association. And so, I think, all these works kind of, have really come a long way in terms of better able to pinpoint what's going on. And lastly, I want to plug my own paper here, which we just got out on medRxiv, which kind of extend this beyond variants and genes, but also looking at gene tissue pairs and basically saying, can we identify not only what the causal gene underlying a given association is, but what is the causal gene in a given causal context. In this case, we applied it to gene tissue pairs from GTEx, and so we have a given gene in 49 tissues, and oftentimes genes between tissues, so a given gene across multiple tissues, are highly correlated with one another, and so we have this additional problem, and what this approach, what we're calling TGFM, is able to do is really tease apart these causal gene tissue pairs from other tagging ones. And here we just have this nice example for an eczema locus, where we're getting the gene OVAL1, which is kind of a known eczema-related gene in the immune-related cell type of lymphocytes with posterior inclusion probability of 0.75. But yeah, and lastly, I want to end it with where this space is at in general, and kind of where we are in the field. And I think there's been a lot of interest and kind of thought on this recently, and I think it's important to consider moving forward. But and so first, I want to reference a paper from Yao et al. Nature Genetics 2020, which they introduced this method called MESC, or MESC, which estimates how much trait heritability goes through disease. And so this is estimating a genome-wide parameter, and that genome-wide parameter tells you how much of the trait heritability, what proportion of it is going through predicted gene expression levels. So if you have a, in this case they use GTEx data, and how much of the trait heritability for a wide range of diseases, which we're showing on the y-axis here, goes through predicted gene expression levels in a whole bunch of GTEx tissues. And their estimate, averaged across traits, was about 10% of disease or trait heritability is muted through adult tissue predicted gene expression levels. And I think it kind of naturally leads to the question, what about the remaining 90%? And so I'm going to propose a couple of explanations, and I don't know what is right, but I am curious for the answer, and I'm kind of actively interested in this question. But the first is that, well, you need gene expression levels in the correct disease context, and potentially bulk, steady state, healthy adult tissue samples is not the correct context for disease. And perhaps you need really fine-grained cell types, and perhaps you need, those really fine-grained cell types, kind of at the right moment in time after eating a big meal or something like that. Perhaps really that's what you need to kind of get at what's necessary. And, so I think there's been, been a fair amount of support for this approach. And, I think perhaps it's a good time to look more into it, and that, there have been a lot of recently generated single-cell EQTL data sets in the past 2 years or 3 years at this point, that have spanned really diverse cell types and contexts. And, I'd be curious, how much these additional, truly context niche specific EQTLs are going to add to our disease mediation heritability. And then second is this approach in this MESS paper looks at gene expression, how much variation in the trait can be explained by a gene going up or down due to a genetic variance effect. And potentially, this kind of one-dimensional gene going up or down is not enough to capture kind of the complexity of biology. And, we have access to many molecular QTL phenotypes, and, I think, assessing what the contribution of these other molecular phenotypes, is of great interest, and, helping us understand what's going on. And then thirdly, there's been another recent paper from Jonathan Pritchard's group, Mostafavi et al., Nature Genetics 2023, which basically showed empirically that GWAS and EQTL studies are differentially powered to detect different classes of variants. And basically, the end message is that these disease-relevant genes are harder to detect EQTLs for at the current EQTL sample size. That's my interpretation of the paper, is that it's really difficult in these important, constrained GWAS genes to detect EQTLs for them, whereas those variants and the variants that the gene effects go through potentially are easier and differentially powered to detect through a GWAS. And ultimately, I guess the thought is, however, I view this as a power issue, is that this will be mitigated as EQTL data sets increase in size and potentially capture increasingly diverse tissues, cell types, and context. But yeah, so that's my brief summary of the state of the field. But in conclusion, I've talked about integrating GWAS and EQTL data to uncover biological mechanisms underlying disease loci. I've talked about some sophisticated methods that have been developed to distinguish these disease causal from tagging genes. And the extent to which EQTL data can explain GWAS loci is currently limited, but will increase as EQTL data sets increase in size and capture increasingly complex cellular context. But that's it. Thank you guys very much.
Anyone has any questions, please feel free to come up to a microphone. I thought I would get us started by asking, if you were going to add in all those multidimensional QTLs available, would you start by first using your EQTL data sets to identify tissues and then layer in the multidimensional information for that cell type or tissue type, or do you do an all versus all mega model at the start?
It's funny you say that. I mean, I'm biased here, but so, I presented this paper, or I advertised for our recent preprint on TGFM, which kind of throws all the gene tissue pairs in together. And we recently got reviews back on that paper, and the reviewer asked, how do you think this compares to an approach, a two-step approach where you first try to identify genome-wide what the causal tissue is, and a second approach.
That wasn't your reviewer.
So I guess my short answer is I hope to have a very clear answer to that question in a couple weeks. But my kind of gut, and I kind of strongly believed that, kind of this integrative approach will ultimately lead to calibrated estimates of posterior inclusion probabilities that the two-step approach could never do. And I think this approach will increase power as well. So, I think in general, if you have the data, throw it all in together and kind of let the model decide what's going on. Yeah.
I have so many other questions.
Sure.
How do you think about how tissues interact with each other then, right? So as you start to kind of look at, if you identified perhaps in an example where there were two different tissues, have you thought about how to, just the same way we look at kind of clusters of gene expression, right, or variants that together co-predict the expression level of a gene or methylation level, is there a way to then extend this to look at interactions between tissues that might result in disease?
I think there would be ways to look into that. I think it's interesting, but I think, at least the way I'm thinking about it, it would be like a statistically challenging problem to do. And like, any type of interaction test is, you need more samples. So I don't know. Perhaps it's, well, okay. So from a genome-wide perspective, if you're interested in, looking across the genome and aggregating signal from everywhere, and not, doing this kind of locus level analysis, which I highlighted in this talk, I do think you can kind of start to look at, kind of pairs of tissues interacting with one another. And I think there probably would be enough signal there. At a locus level, I think it would be challenging. Yeah.
Hello, thanks for a very clear talk. I was just wondering the extent to which there is a very active research program which explicitly picks up the results of studies like this and continues them down the funnel to try and really nail, is this doing what we actually think it's doing? I mean, obviously this is a step after GWAS to understand more about the biology, but there's still more to be understood, and like, how do these studies get picked up?
Right. So you're saying like, these studies are making predictions, and what is happening with these predictions?
Right. Thank you.
I'm not sure I have a great answer to that. I'm not entirely sure. I mean, I guess the way I see it happening is some type of, collaboration from, a lab like, Alkis's group, or, with me, and that, we're generating predictions, and, some lab that, has the infrastructure to experimentally validate what's going on and really get at how good these predictions are. I mean, we can, make all the simulations we want and, say that we're identifying, the causal gene with calibrated posterior inclusion probabilities. But, I think it's all kind of out the window when you start talking about real biology. So I don't know. I mean, I think it would be great. I mean, I'm not an experimentalist. I wouldn't even know how to, what experiment to begin with in terms of trying to test this, but I'm sure there's someone here knows. Yeah.
We have two questions online. The first is, how do these models how well do these models account for infinitesimal effects from other variants? And could these spurious effects be another reason for the model not performing well?
Can we clarify whether it's infinitesimal GWAS or EQTL effects?
Choose
Okay.
your adventure.
Sure. I would assume it's EQ. I mean, if it's GWAS effects, I mean, there's no real concern there in that, like, I think if there are infinitesimal GWAS effects, there's no concern. I mean, the TWAS probabilities or the TWAS p-values are going to be well calibrated, even if there's infinitesimal GWAS effects. If it's EQTL effects, it gets a bit more complicated in that they're going to be hard to pick up from your predicted gene model. I will advocate that our recent fine mapping paper, TGFM, models uncertainty in genetically predicted gene expression by assuming some posterior probability distribution on the causal EQTL effects. Instead of treating them as a point estimate, we basically account for this uncertainty in the prediction model and the ultimate posterior inclusion probabilities are outputted. In addition to that, there's been another recent Nature Genetics paper that just came out at the same time as the causal TWAS paper called GIFT, so I don't remember the first author, but its package is GIFT, 2024 Nature Genetics. And that approach, they also basically do this uncertainty integration of uncertainty in the causal EQTL effect sizes in the outcome. So in that case, I think it becomes a power issue in that you're going to be underpowered. So if you, I guess short answer, if you have the right model to deal with uncertainty in causal EQTL effects, it's not going to be a false positive problem. It's going to be a power problem. And as you get to a more polygenic architecture, it's going to be harder and harder to really detect these things. But I don't think you're going to get increasing false detections, which is kind of the ultimate concern, I guess, in a statistician's eyes.
Thank you. We have another question. How much have methyl QTLs and other epigenetic QTLs contributed to the understanding of GWAS loci?
To my knowledge, there hasn't been a whole lot of work on it. I guess I'm from this really biased space of really focusing a lot on EQTLs and stuff like that. And I haven't heard too much about it. I mean, I think it maybe comes down to having kind of the right methylation QTL data set and, high enough sample sizes in, interesting enough contexts, which I do not know the answer to. But, if you have it, I don't know, run it. I mean, these are quick to run. I mean, but yeah.
And then we have one more question online, and I think it's related to the one that was asked in person. Are you aware of any databases where people can find the results of these GWAS and TWAS studies so that we can go and dive into the biology?
So I mentioned Sasha Gusev a lot. He did the initial TWAS paper, and his software suite is called Fusion. And if you go onto the Fusion webpage, there is kind of a detailed list, I believe, of all the current TWAS or like some organized run of TWAS associations across a range of UK biobank traits using GTEx EQTLs from, I kind of forget how they did the gene models, but I believe there would be my best guess as to where you could find a synopsis of these TWAS associations in diverse traits and tissues.